In this short video, we're going to talk about some of the tools we can use to gather information that will help us put a potential functional name on our assigned protein sequences. So we've already seen how we could use BLAST as a way of finding an outgroup sequence from the human louse genome to use with our assigned bird lice ecomorphs. And we're going to go back to the BLAST search results because there's actually a lot more data there. So here we are at the BLAST output page, and we've already have learned how to interpret data here and look for sequences of interest and how we're looking at sequence alignments. Okay, but I'm going to scroll back up to the top, and you'll notice that when this will not always happen, but hopefully it'll happen for you, is that it will also show you if your protein has some conserved domains that have been seen before in proteins of known function. So you can actually click on this little picture here, and it will take you to then a new site called the Conserved Domain Database. So we're going to do that now. And it will bring up some more details about these models that our protein seems to be showing some similarity to. So it shows us visually kind of where these similarities are, and if you put your cursor over them, they'll give you some more information about them, okay? But they also have a list here, and you can expand on that. So here is a hit to a protein family model, and we're going to talk about the PFAM database in just a minute, but one called ATP synthase subunit C, and if you go back to the BLAST search results, that actually was what most of the hits we see to our protein sequence are called. All right, and here it potentially tells us a little bit more about it. It shows us how well it lines up, and in this case has a pretty short little descriptor. Here's a different set of models of protein domains, and here it tells us a little bit more, that this appears to be similar to a model for a ATP synthase subunit that's called a proteolipid subunit. That starts to make me think that it might be in the membrane. All right, it tells us that these subunits are found in eukaryotes and have members from fungi, plants, etc. Okay, so I'm starting to actually get some idea of what maybe my protein is doing. So this is one place we could go to capture that information. Another place we can go to is actually this PFAM database, and so we're going to go there separately from its connection to BLAST. So I brought up the PFAM homepage, and PFAMs, as it says here, are a large collection of protein families that are built off of multiple sequence alignments of proteins of known function. And then they make a model of that, and then you can put in your sequence and see how well it matches to a model. And as you can see, they have a lot of different models of protein domain families. So if we put in our sequence here, we can send that in. And for our sequence, this is the hits we got back. And again, it says ATP synthase subunit C, which we saw before from the BLAST. We can see how well it aligns. So we can see it's in pretty good alignment here. It gives us some of the details. But we can also go and get a little more information about this protein domain family by clicking here. And this is usually given to you in the format of like a Wikipedia page, but has references to the literature, has sometimes pictures as we see here. And this can be really useful to you to learn something about your protein of interest. All right. So again, ATP subunit C tells me some more about it, what kinds of jobs these ATPases do. It starts to tell me here more about my particular subunit. It tells me it's in the so-called F0 or V0 complex, which is the complex that's actually in the membrane. So I've already now have hypothesized my protein most likely sits in the membrane, and that's the last piece of data we're going to gather, which is to make use of some tools that allow us to predict where in a cell a protein goes. So we're going to go to a collection of tools and interpretations of those tools called psort. And this is one version of psort that specializes in animal and fungal sequences. And so I've selected that we have an animal sequence here, put in my sequence of interest, and I'm now going to ask it to look for signals in this protein that may tell us something about where the protein goes. So I'm going to go ahead and send that. And the data I got back is at first a little too summarized. I may not totally know how to interpret this, so I'm going to get a little more detail. And the details, it tells me who are some of the proteins in a very specialized and well-documented database called SwissProt. Who are some of the proteins my protein looks like? And notice many of these say integral membrane protein. That kind of agrees with what we were seeing before from our PFAM hits. And it gives me some other things that I'm not quite sure how to read. But up here it says, show me some of the traditional prediction data. And that's really what I want to get to. So I'm going to go there. And now it starts to walk me through some of the analysis it did. It has a few ways of looking for signal peptides, which if you remember from cellular biology, are sequences that basically say to the cell, hey, put this protein into a membrane or across a membrane. One of them predicts a potential cleavage site. Other ones suggest there may not be a signal peptide. And one of the things you'll have to prepare yourself for is you're going to have to basically sort of draw your best conclusion from some data that might have some contradictions within it. It does, though, predict that there are transmembrane domains. It finds 5 of them, and it tells me where those domains are. Again, that agrees with my PFAM hit. It can tell me kind of how that protein is sitting in the membrane. And it tells me that it does not look like it's a protein that goes to the nucleus. Well, if you're in the membrane, you're not going to the nucleus. It looks to see if it has any other targeting signals to it that might tell it where to go. It doesn't find any of those. And so in the end, what I'm left with, and this is in agreement with my initial blast hits, is I have a protein that appears to be in the membrane, and I have hits that tell me that it most likely is part of some sort of ATP synthase complex. So I now can start to put a name on it, and I can even go to the literature and start to learn more about this protein.